So first of all, I just want to tell you that I'm not affiliated with Snapgene in any way or form. I just bought this program for myself because I find it particularly useful in the projects I'm doing, especially in designing my cloning reactions and designing my PCRs. Of course, I use it also to view and edit DNA. I'm going to show you some how to use some basic features of Snapgene. And for this, I'm going to use the Snapgene Full. So this is the Snapgene Full program. Snapgene also has a free version, which is called Snapgene Viewer, that I'm going to talk about a bit later. First of all, what I'm going to show you? I'm going to show you how to open and view DNA files, and of course view the sequences and what features are and so on. Number two, I'm going to show you how to view restriction enzymes and use different enzyme sets. I'm going to show you how to simulate an agarose gel electrophoresis. For hands-on real practice, I do have a video on that. Link will be in the description. Number four, I'm going to show you how to add primers and simulate different PCR reactions. I do also have a video about that regarding hands-on. I'm going to show you how to simulate mutagenesis, and of course I'm going to show you how to simulate cloning reactions and basically view the history of your cloning in different text and images and how to export them and so on. Again, in the description will be available a couple of videos that I have regarding hands-on on how to make a restriction digest and how to do a gel agarose extraction, both very useful for cloning. I told you earlier that Snapgene also have a free version, which I would highly suggest to download. It's called Snapgene Viewer. Link will also be in the description, and I would suggest to try the free trial of Snapgene. So basically, if you put your email in there, you can try Snapgene for 1 month. It's for each email you get a free trial, if you catch my drift. Okay, so without further ado, let's go into how to open and view a DNA file. So before viewing a DNA file, I'll need to download one. I'll download the plasmid from Adgene, exactly the sequence, which is PUC18, for example. So this is the PUC18 plasmid. We can view sequences, and from here we can download straight to Snapgene, the file for Snapgene. As you can see, this is the PUC18 plasmid from downloaded from Adgene. So first of all, you have the plasmid map. Then you can view the sequence, the exact sequence of the plasmid, which with the features. Then you can view and choose your enzymes. These are the features that Snapgene automatically annotates. These are the primers, and this is your history. We'll go into that a bit later. First of all, if you press on a feature, it shows you exactly what it does. For example, this is, if you double click on it, it will show you. So this is the gene for beta lactamase. This is actually the ampicillin resistance gene. If you hover over them, it's going to show you, for example, the LAC-Z gene, which is LAC-ZA fragment, or let's click on this operator. So that's what I like about that gene, is that it tells you what this particular feature does. Let's see here. So cap binding site, LAC promoter. Or this one is a high copy number, origin of replication, and so on. So this is how you download and view the map, the sequence, the enzymes, and the features of a plasmid or a piece of DNA. Okay, so this is how you download and view your particular DNA. You can see here in black the enzymes that cut this particular piece of DNA. Let's go to the multiple cloning site, because this is where you are usually gonna do your cloning, and this is where you're gonna clone your gene of interest, right? Let's go to sequence and see in more detail. So if you hover over XBA1, for example, it's gonna show you the cut site, and it's gonna show you exactly the overhangs that it's gonna leave. And this is true for all enzymes that cut here. Let's, for example, go a bit further down. This one is an enzyme which doesn't leave overhangs. This one the same, and so on. For enzymes, for choosing enzymes, for example, just showing the enzymes that you want to be presented on the plasmid map itself, you can go from here, and for example you can choose unique and dual cutters. So this way the program will show you both the unique cutters and the dual cutters. If you go to map, see, it shows you all those enzymes. You can also choose from here in the menu. I'm using Mac, but you can do the same thing in Windows. Let's go to use enzyme set, for example, the enzymes from New England Biolabs, right? And it's gonna show you the enzymes from NEB. Or let's choose all commercial. This is going to show you all of the enzymes that cut here. And if you, for example, choose an enzyme, let's just search for NDE1, and it's going to show you that particular enzyme, and it's going to show you the particular position in which it cuts, right? If you select it from here and go to map, it will take you directly to it. Okay. You can also, for example, show or hide enzymes. So let's hide all enzymes. In this way, it will give you, the program, it will give you a clear map without the enzymes. Let's just say we want to show one enzyme in particular, which is going to be a unique cutter, and I want to see just eco R1 on the map. I'm going to select it from here, and then you can see that it's going to show me that particular enzyme. What I can do also is, if I right click, I can copy the recognition sequence, I can hide the enzyme, or I can highlight the enzyme site, so it will show me, for example, the enzyme site in highlighted in yellow. Yeah, so I would suggest to play with the enzymes and show on, because you can do all sorts of things right here. You can, I don't know, for example, type 2S nucleases is going to show you exactly those types of enzymes that cut. Yeah, so this is how you choose your enzymes, and how you can basically do and use enzyme sets here. So now for simulating an agarose gel, let me just open another plasmid, right? So let's just go File, Open Files, And I'm gonna choose another plasmid, so another sequence. Let's just say I'll choose Cindy CFP. So this is the sequence map of P Cindy CFP. This is a beautiful plasmid, so let me show you some images with it. So I have this plasmid and I also have PAC18. I'm gonna have to go to Tools and simulate an agarose gel. This is gonna be PAC18. So let's say, for example, I like to cut with EcoR1. This is the piece. This is the sequence, and this is cut with or amplified by PCR. But let's just not worry about this right now. So cut with EcoR1. You can see here on the simulation that is gonna give you a fragment that's gonna migrate exactly as the PAC18 linearized fragment, because you're just cutting here at one site, right? And it's gonna migrate just below the 3000 base pair mark of the ladder. So as you can see here, the first lane is the DNA ladder. You can choose the ladder from different suppliers. So let's just say edit marker list. You can see here there's plenty of suppliers to choose from. So me, for example, I chose as the default marker the one from New England Biolabs. 1 kb plus DNA ladder from NEB, because that's what I'm using in my experiment. So this is the ladder I have, but you can choose whatever. Let's go back to lane 1, which is the sequence of the PAC18 plasmid. So I'll cut in one place with EcoR1, and let's just say I'm gonna cut in different with a different enzyme, XBA1. As you can see, it's gonna show me on the gel the actual fragment, so this one, right, which is about 2700, and one that is off the gel of about 27 base pairs. But let's say I want to add to my agarose gel Cindy also, and let's say that I'm gonna digest exactly with the same enzymes that I digested PAC18, so this will be EcoR1 and XBA1. As you can see, this is gonna give me two fragments, one from the cut sites here, so it will be XBA1 EcoR1, and the tiny fragment that contains the CFP gene, right? So the cyan fluorescent protein gene. Of course, I can change the number of lanes here. For example, I have 15 lanes or 16 or whatever. I'm choosing 10, and I can also choose the percentage of agarose that I'm using. Let's say I'm using 2.5%, but I'm mostly using 1%, so this will be my gel. You can print gel and copy, export gel data, and so on. And you can also edit some gel preferences from here. Okay, so this was the agarose gel simulation, which again you're gonna do from Tools, Simulate Agarose Gel. Adding primers, I'm gonna use the same sequence, PAC18. And I'll go to sequence. As you can see, some primers are already added here, but let's say that I want to amplify this gene, right, with its promoter. So I want to amplify the ampicillin resistance gene with the promoter. So first of all, I'll need to choose a primer. Let's say that I want to go from here, starting with the G. As you can see, it shows me how many base pairs I've chosen and the melting temperature of the primer. So let's say I'm I want a primer of about 25 base pairs with a melting temperature of 52-53 degrees Celsius. So I'm just going to select this piece of DNA. I'm going to go to primers, add primer, and it's going to prompt me to choose top strand or bottom strand. Because this is the forward primer, I'm going to choose forward strand. I can name it from here. So let's say amp forward. And I can choose the color of the primer. So let's make it purple, right? Let's make a purple primer here. You can also choose to add the reverse complement, and you can choose the hybridization parameters from here. So how many bases should it match? What melting temperature should be required for this specific primer, and show additional binding sites at the sites that do not match. So basically, this also shows you the additional binding sites for this piece of DNA. Right now, I'm not interested in that, so I'll have a primer which is about 25 base pairs long, all anneal, and it has a TM of 52 degrees Celsius. So I'm gonna just add this primer. And you can see here in purple with its name, with one binding site, and it's going to also show you the CSG content and so on. Now I'll have to choose the reverse primer also, so let's just say that I'm starting from here. Remember that the forward primer has a melting temperature of about 52 degrees Celsius. I'm going to select the piece of DNA that I want until I get to 52 degrees Celsius. This is gonna be about 17 base pairs. I don't want that. I want to have at least 18. Okay. Let's just say this is my primer of about 53. I'm gonna go to primers, add primer, and now I'm gonna choose bottom strand because this is the reverse one, right? I'll call it the reverse. I'll choose as color, I don't know, let's say green. Let's say that all reverse primers are in green. And I'll add the primer to template. As you can see, I have 25 annealed bases at about 53 degrees Celsius. Okay, so now that I added my primers, which is this one, I can also see them in maps. Let's hide the enzymes for now to see the primers better. And I'll have this one, amp reverse and amp forward. If I press Ctrl and click them both, it will show me the region that will be amplified. And okay, let's amplify this region. For this, we'll need to go to Actions and do a PCR. Okay, I'll choose the primers that I want to PCR from here. So first of all will be the forward primer. Then I'll have the reverse primer, and as you can see, it will amplify a region of 1045 base pairs, and I can name it from here, amp R gene, and then do a PCR, and this will be my PCR fragment. Of course, I can see the map, I can see the enzymes, I can see the sequence, I can see the features and the primers that anneal to this particular piece of DNA that I amplified from my plasmid. What I can also do is go to primers and just hide primers. So hide them all. I can import them, I can export them, plenty of things I can do, right? So this will give me a clean map of my piece of DNA. I can also basically hide the features from here and show the features this way if I wanted to. I find this particularly useful when I try to do site-directed mutagenesis. What does this mean? Let's say, for example, that this is the sequence, right? And I want, let's show the enzymes here. Okay, let's say, for example, that here there's an enzyme, and I want to remove this restriction site from my plasmid. What I'm gonna do right now is I want to just replace a nucleotide here with another one, again to remove the restriction site. What I'm gonna do is, for example, I'm gonna choose a primer here for site-directed mutagenesis. Let's just say this is the primer. I'm gonna do top strand, and I want to exchange one base pairs from here. So for example, to eliminate the recognition sequence, I want to change the G from right here with the T, right? So this one is that, so we have G, T, C, 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 and so on. I'll want to exchange the G with a T. I'll put it in a capital letter. So this will not anneal to the template. I'm going to add the primer here. And of course, I'm going to need to add the reverse complement for site-directed mutagenesis. So I can just, add a primer, not like that, sorry. I can select this primer also. I can copy it. Primers, add primer. I'm gonna paste it, but I'm gonna paste the reverse complement, right? So this will be reverse site-directed mutagenesis primer. Okay? And these primers will be complementary to each other. And okay, how will my plasmid look without the G and with the T inserted in here? Let's go to Tools, Actions, Mutagenesis, and I'm gonna use this primer, reverse site-directed mutagenesis, to do an exact version of POC18 minus that G from here, which I'm going to replace with an A. Okay, so ready to do mutagenesis. One base pair will be deleted, one will be inserted, and I'm going to call it POC18 minus hind A3. I'm going to mutagenize, and I'll have this plasmid right here. And if I'm going to go search, because this is the mutant version, right? And if I'm going to search for enzymes, let's say that I'm going to go to enzymes, and then I'm going to look for this enzyme that I deleted, right? And I'm going to add one chosen enzyme does not cut. And this is going to be my enzyme that I've eliminated through site-directed mutagenesis. So that's why I find it particularly useful to simulate mutagenesis using a single primer. Let's say we want to clone a gene into this plasmid right here. So this is a mammalian expression plasmid, okay? This is used to express foreign cells in mammalian cells, for example, HEX or HeLAs or something like that. As you can see, this is the promoter. This is the enhancer right here, the CMV enhancer and the promoter. So first thing I'm gonna need to do is look for the multiple cloning site in here. As you can see, these enzymes right here are this is the multiple cloning site because it contains many enzyme sites. And let's go to the sequence and have a look at it. Okay, let's say for example that in my lab available for me are BAMH1, so this enzyme. So I'll have this enzyme, I'm gonna highlight it, and XBA1. And that means that I'm gonna clone my insert or the gene that I want to express between this restriction site right here and this one. So I'm gonna clone it and replace this piece of DNA right here, the highlighted one. Well, what I'm gonna clone into this this plasmid. Let's say for example that I want to clone the Cindy CFP gene. What I would do is basically amplify this gene from this plasmid right here with primers that contain the restriction sites that I want to use. So let me show you how this goes. So let's go to sequence. Okay, this would be the start of my gene. This is the start codon right here. So I want to pick a primer. Let's say I'll make it 53 degrees Celsius. So the TM is 53 degrees Celsius. I'm gonna add the primer and I'm gonna say top strand, of course, because it's the forward one. So I'm gonna say forward CFP. Let's add the primers. The primer. What I'm gonna need to do is basically at the end of this primer right here, at the 5' end, right, I'm gonna have to insert a restriction site so that my cloning will work. And because you can see this one is the first one, BAMH1, then it's the XBA1 here, I'll need to introduce BAMH1 into my forward primer and XBA1 into my reverse primer. So what I'm gonna do is just copy the recognition sequence from right here. I'm gonna go to my primer, then I'm gonna edit it. At the 5' end, I'm gonna paste my restriction site. Then I'm gonna add some nucleotides because, well, the enzyme actually needs some couple of base pairs to just to come and cut at this DNA site, right? So okay, I'll just add this primer for simulation sakes, and this is basically 53 degrees Celsius, right? Sorry, this is my kitty right here. Then I'm gonna go at the end of my protein right here, and I'm gonna add the other primer, again 53 degrees Celsius right here. So primers, add primer. This one is the bottom strand because it's the reverse one, and I'm say reverse CFP. Okay, then let me check out pcDNA 3.1. So I'm gonna have to insert this this enzyme site right here. So I'm gonna copy the recognition sequence. I'm gonna paste it at the end of the primer. As you can see, here is from 5' to 3'. Here's the reverse. And then at the end, I'm gonna insert some random bases, let's say AGT or whatever. So this is 52 degrees Celsius, this will be okay. I'm gonna add this primer to template. What I'm gonna need to do next is simulate a PCR reaction. Well, actually, this is what I'm gonna do in real life. So I'm gonna do a PCR to try to amplify this gene right here. So this would be the forward, this would be the reverse, this I'll call CFP gene, then I'm gonna PCR, right? And this is what I'm gonna do is I'm gonna extract this piece of DNA from the gel. When I do my cloning, I'm gonna cut with BAM-H1 and with XBA1. And for the simulation, what I'm gonna do is go to Action, Restriction and Insertion Cloning, Insert Fragment. As the vector, I'm gonna choose pcDNA. I'm gonna cut this one with BAM-H1 and XBA1. This would be the region to replace the small one, 62 base pairs one. As the insert, I'm gonna choose, of course, my amplified product right here, which I'm also gonna cut with BAM-H1 and XBA1. Okay, so this will be my fragment that I'm gonna insert, and Snapgene is gonna give me the product. This will be my final product. As you can see, I have the CFP gene. This is the polyadenylation sequence, and this is the promoter and enhancer. So this is my clone that I'm gonna use to transfect mammalian cells with and make them glow cyan. I can just rename it here. So let's say pcDNA CFP. Okay. I'm gonna clone, and this is gonna be my my final product. Of course, I can save this and whatnot, right? And if I go, let's say for example, to simulate an agarose gel, and I'm gonna cut this one with XBA1 and BAM-H1, I'm gonna see those two fragments. This one that I inserted and this one, which is the main plasma. Okay, so pardon the noise. It was my crazy kitty. Hopefully this has been useful, and well, if you liked it, just give feedback on what you want to see next.